Good afternoon, everyone. My name is Joana Damas, and I work with Harris Lewin. We requested me to introduce you to today's speaker, Professor Leif Andersson. He is a specialist in genetics and genome biology, and he's been studying for the last 30 years mostly domestic animals, trying to understand the relationship between genetic variation and phenotypic variation. But also, in the last years, he started looking also into wild populations that we will talk to us today, including the Darwin finches, the Ruff, and Atlantic herring. He is currently a professor of functional genomics at the Uppsala University, of animal genetics at the Texas A&M University, and also a guest professor of molecular animal genetics at the Swedish University of Agricultural Sciences. And amid all the recognitions he received during his career, he is nominated as a member of 3 major Swedish academies, namely the Swedish Royal Academy of Science, the Swedish Royal Academy of Agriculture and Forestry, and the Royal Physiographic Society in Lund. He is also a foreign associate of the US National Academy of Sciences and an honorary member of the American Academy of Arts and Sciences. And also during his career he received numerous prizes, between them the Wolf Prize in Agriculture that he received in 2014, and just last year he received the Linnaeus Medal. So please join me in welcoming Professor Leif Andersson.
Thank you very much for the nice introduction. In fact, just need to make one correction. So in fact, I started by working on natural populations, and that was the herring which was seen here. But when I did this first work, I found out that we didn't have any resolution to really study the population genetics and evolution biology of the herring, because the technology at that time, that was when I was a graduate student in the 1970s. So I left the natural populations for 30 years when I was working on domestic animals. But when new sequencing technology was introduced, which opened up the field of biology, that you could study any species of interest with an interesting question, and herring is one of them that I'm now working with. So another thing I wanted to talk with you about today is what type of genetic changes are most important for evolution changes and ecological adaptation. And I think that the work we have been doing, because they're both in domestic animals but also in natural population, is both that the regulatory changes and changes in protein sequence are important. Because this has been the discussion. At one point we thought that everything was happening on the protein level, and then the pendulum shifted towards that everything is regulatory, but I think it's very clear that changes occur on both levels, and particularly our herring data really point that adaptation in the herring involve changes in the protein sequence. And then I also say that we both see sequence changes and structure changes. Sequence changes could be single nucleotide changes, and which of course will be many more numerous than structure changes, but structure changes can have important impacts on evolution. And I will give examples of that as well. And then I would like to mention this concept of evolution of alleles haplotypes, which I think is the norm in natural populations, that when you see selection, you often select on more than one mutation. There could be one mutation that starts a selection process, but that is then followed by additional mutation accumulating, affecting the same gene or same haplotype. So I would like to illustrate that, because this is something I've observed in domestic animals. And which is interesting, because the history of domestic animals is only 10,000 years. But we have several examples where we see that the phenotype is determined by the combined effect of 2 or more mutations. And one of the examples is change in coat color in the pig. So this comes from the work we did when we crossed the wild boar with the domestic pig. And what we found was that there is a mutation in the KIT gene, which is a tyrosine kinase receptor, has a critical role for the migration of melanocytes. And in pigs, you have a duplication, a 450 kb duplication that involves the whole coding sequence, all the coding exons, but also up to 100, 150 kb on each side of KIT. But what this is that the regulatory element for the KIT gene is widespread, hundreds of kb outside. So the duplication doesn't contain all the regulatory elements. So that means that at least one of these copies is dysregulated. And that leads to confusion because KIT signaling is critical during the migration of melanoblasts from the nerval crest out to the skin. And you see this guy. So here in this area, the melanocytes made it to the skin, whereas in this area, they're lacking, in the white area, they're lacking skin. So this mutation hit first. Apparently, some humans liked it, kept these pigs, and then 2 or 3, 4 mutations happened. So there is an additional set of duplications. But then there is a splice mutation. That splice mutation leads to exon skipping, and that exon codes for the tyrosine kinase domain, which is absolutely critical for KIT function. And that means that these white pigs, they have 2 copies of KIT, one normal, maybe dysregulated in the expression, and a second copy that is combined ligand but cannot signal. The combined effect of regulatory mutation plus change in the coding sequence gives this strong phenotype. So this is the strongest KIT allele which is viable that has been described in the literature. And the reason why it is so strong, it's combining regulatory and coding changes. So what you have here is that if you combine the wild type allele with a splice mutation, that will give in the heterozygote a mild phenotype, but it will be recessive lethal because a vertebrate cannot live without KIT signaling. And if you knock out the tyrosine kinase domain, there's no signaling. But when the splice mutation hits the patch, then you get the viable allele giving dominant white color. So the effect of the splice mutation is completely different if it occurs on the wild type allele or on the mutant duplicated chromosome. Because if you have the duplicated chromosome, they will always have at least one functional copy per chromosome that can contribute to tyrosine kinase signaling. So what we see in the white pigs, in fact, so these are widespread. Humans have been selecting them for hundreds of years. And what we see, they have a slight reduction in red blood cell because KIT is also important for hematopoiesis. And that's why it's lethal if you completely knock it out. But I think this is an important concept, that the consequence of mutations is very different when a major causal variant has hit one gene. So it changed the game at the KIT blockers. So today I will give you 3 examples. So I will start with the genetics of Ruff 6, which I think is as exciting as it sounds. Ruff is this bird. So this is the 2 males. They have these beautiful ornamental feathers, enormous. I hardly know any species that is so polymorphic in its pattern. And they're only this variable during the breeding season. Outside the breeding season, all males look the same. But this is the fascinating thing with the Ruff. There are 3 different types of males which are genetically determined. There is the independent male, which are like 90% of the population. At the breeding time, they have these beautiful ornamental feathers, which could have all type of colors, red, black, they could be striped, they could be barred, they could be spotted. So almost everyone has its unique. It could combine. So they have head tufts up here, could be different from the color down here. So the whole thing about the Ruff, which is why this has evolved, is in the breeding season they have leks where the independent males fight and defend territory. Like 1 square meter, where they invite females to come and where mating takes place. Then you have the satellite male, which is distinct, slightly smaller, always light ornament, often white or light-colored ornamental feathers. They do not fight, but their strategy is to allow these guys to beat them up like this. So this one peck on this one, and that attracts females. And when the male do this, he gets access to the mating ground and have a chance to mate without needing to defend territory. The third type is this guy, and he's a female mimic. Maybe 1% of the population, smaller, look like a female, and sneak around on the mating ground and try to get matings. So the amazing thing is that these things is determined by so these are determined by dominant alleles at a single locus. So this is this enigma. So how can such complex phenotype have such a simple genetic inheritance? One locus on one chromosome, and it influences body size, behavior, plumage color. And for geneticists in the golden area of genetics, this is a challenge to try to find out what it is. And for you to really enjoy what that is, I should show you a video taken in Karesuando, which is on the border between Sweden and Finland, in the far north, about the polar circle. So it doesn't want to, don't get the. What do you say? I don't get the cursor, that's the problem.
I can't see it on there. Let me see if I can.
Okay, I see it
got
there.
it?
Okay, now I see. Okay, fine. Okay, so here are the male Ruffs flying in. This is a satellite. This is an independent male. You see, this is red with some spots here, and black up here, and carotenoid pigmentation there. This is the satellite with its white ornamental feathers. And this is the feather now. And you see, it's basically impossible to see if it's not a female. So this is how they fight. To defend territory. So these are independent males fighting to establish territory. Now they have established territory. They are standing on their post waiting for the females to arrive. But this is this other guy, you know? This is a satellite. He doesn't defend territory. The other males recognize that he's different. They don't attack him. They allow him to go around feeding. Now the females come. And now the action starts. So now this guy invites him to start this ritual fight, where he will lay down, and female watching, and look. Female is ready to be mated. And there is some things going on there. And the satellite mates this time. And now you have to watch, because here is the feather, the female mimic male coming. These other males will start fighting each other, and he takes his opportunity while they are busy fighting. So what is the genetic basis for this? So how could this be a single locus, two dominant alleles? And what we did was to do a genome assembly and then whole genome sequencing. So the reason why we could do that there was a PhD student that did a project on this in the late 90s, or during the 90s, I would say, spent 5 years watching this, collecting blood samples, and we dig them out of the freezer, prepare DNA, run the whole genome sequencing, and then we compare FST between the independents and the satellites, and you get this beautiful signal. So this is just noise on the other scaffolds, but here is one genomic region, 4.5 megabase region, highly significant differentiation between the groups, and consistent with a single locus. So what's going on? Well, it's an inversion. So it's a 4.5 megabase inversion which have happened here. So it's about 100 genes, and so I will come back to that, but the satellite and feather has exactly the same inversion breakpoints. And I will come back to what the difference are between the two chromosomes. But what is worth noticing here is that, so for inversions, it's very important what happens at the breakpoint, I would say. And what you could see here is that this breakpoint disrupts centromere protein N gene. So when the inversion flips, that gene is broken. This gene is essential for life. So if you knock out that in somatic cells of humans, they stop dividing. So this is a recessive lethal, which in fact has been confirmed in some pedigree studies of these birds. So this means that the satellite and feather chromosome must be maintained by balancing selection to compensate for the loss of the recessive homozygotes. Otherwise, it should go away from the population. Then is the question, how old, when did this happen? So what we learned was that if you look at these regions which are most divergent here, there's a 1.4% sequence divergence. If you recall that the difference between human and chimpanzee is 1.2%. So these chromosomes are more divergent than human and chimpanzee chromosome. So substitutions rate are different in birds than in humans. So we estimated that the inversion happened about 4 million years ago. So this recessive lethal has been maintained by balancing selection for a longer period of time than the history of modern humans, and which is amazing. And I don't have time to go in because I wanted to cover other aspects as well. So this is like the summary of the evolutionary history of the Ruff supergene. This is often called supergene, where you have an inversion keeping together a number of. So what happened was that the inversion happened something like 4 million years ago, and then a number of changes happened over time, so they evolved into a distinct chromosome here. But then there was a second recombination event which took place about 500,000 years ago, where the wild type and the inverted chromosome recombine. So you need double recombination within the inversion to exchange sequence between them. So the so the feather chromosome seems to be an intact inverted chromosome, whereas the satellite chromosome is a mix of the satellite and independent chromosome. But that happened 500,000 years ago, so they have accumulated new changes after that, and that is what you see here. So this is the region which is contained like the feathered-like organization. This piece came from the independent 500,000 years ago, and you see this. It must have been at least 2 events that created this recombination. So I think this is a very important concept, that inverted chromosome can evolve by recombination, although recombination are very rare. So the problem, of course, is you find this. So inversions are lovely in a way. It's easy to find them, but you have this huge problem to see what are the critical genetic changes, because you have these 100 genes and complete linkage disequilibrium essentially across this. But we had some signatures of selection that I think, and our 2 best candidates are HSD17B2, which we have mutations both in the satellite and the feather. And why is this interesting? Well, the signals we see is that we have 3 deletions in evolutionary conserved sequences flanking HST17B2, which suggests altered expression. And why is this interesting? Well, this is one of the key enzymes in the metabolism of testosterone. If this enzyme degrades active testosterone, and what we know is that these satellite and feather males has lower circulating testosterone and an altered behavior. So it's easy to think that upregulation of this enzyme, at least in certain tissues, would change the testosterone metabolism in the feather and the satellite. The other very strong candidate gene, so if you talk about pigmentation, you could always guess that it's a change in MC1R, and MC1R is located within the inversion. The satellite, which has this white plumage, they have 4 missense mutations at conserved site in MC1R, and we think that is contributing to the phenotype. So I think this is a good way of thinking about how evolution works. So the inversion happened about 4 million years ago. After that, mutations that accumulate on the inverted chromosome that increase the fitness. So I think the inversion itself created a primitive alternative morph, and that had been improved by new mutations, selection going on over millions of years. And MC1R is probably one of the genes. HST17B2 is another gene which have accumulated changes like that. And of course, you also will have mutations on the independent, because the independent has changed behavior. They tolerate this and have developed this behavior to fight, or this, what you say, ritual fight with the satellites. So I think this is what I would like to illustrate with that mutation will have different effects depending on which haplotype they occur. And it's very clear in this situation, but I think the same thing happened on other cases, like the white pigs. Okay, then let's shift to another bird, which are the Darwin's finches, which we have been working, and the reason why we've been able to work with them. So for a long time, it was very embarrassing for me. You know, we do this work on Darwin's finches, and I've never been to Galapagos. But we have been working with Peter and Rosemary Grant that did field work every year for 40 years. And they collected, I think, approaching 6,000 birds they sampled blood from. And in fact, we have all those samples in Uppsala now, and on the way to sequence a good fraction of those 6,000 birds to study their evolution and so on. But we have, so here is the archipelago of Galapagos with the different Isabella here, Santa Cruz, and San Cristobal. So it's a university on San Cristobal, in fact, where we did our lab work when we were there last year. And in fact, one of the Darwin's finches is found on Cocos Island, which is up here, closer to, so it belongs to Costa Rica, in fact. So we have sampled all the species of Darwin's finches. So when we started, there were 14 species on Galapagos, 1 on Cocos Island, and 2 close relatives. But due to our work, we have split some of the species. So we think it's now 18 species, on, including the Cocos finch. So one of the things we have been doing with this data is that we identify genes controlling beak shape and size and their importance for genetic adaptation. And so to identify a gene affecting beak shape, we did a genetic screen where we looked 15 kb windows across the whole genome. So we did whole genome sequencing, individual birds, and we compared those which had blunt beaks. So this is the Magnerostis, the large ground finch with its very blunt beak. This is the large cactus finch with its blunt beak. This was considered to be the same species, but they differ in beak shape. And this is the Difficilis, which has a pointed beak. So this is the pointed type, and this is the blunt type. And then we compare allele frequencies across the whole genome. So what you need to know is this is a recent radiation. So the ancestor of Darwin's finches arrived on Galapagos about 2 million years ago and had diversified in these 18 species since then. And in particular, these are all ground finches, and they have radiated even later, and there's a considerable amount of gene flow between them. So it's a lot of shared polymorphisms. So we were looking for what is distinct between them if you sort them according to a phenotype. And we did that and found which really looked like a non-random pattern where we have a number of what looks to be signals that distinguish those with blunt beak from the pointed beak. And we're convinced that this is a polygenic trait, as you expect, but we found some striking hits, I would say. What was interesting was that 6 of these regions contain genes which previously have been associated with craniofacial development or beak development. And the strongest signal we observed was for ALX1. So ALX1 is a homeobox transcription factor which has a very well established role in craniofacial development. So if you do knockouts in zebrafish, you get a very severe defect in craniofacial development. There's also a syndrome in humans where you get cleft palate and defects if you have a loss of function on one copy of these guys. So we look at the haplotype data of this and saw that we have a strong linkage to this equilibrium over 240 kb. And in this region, we could sort haplotypes according to which type of beak. So here you have different species here. So these are the haplotypes coming from blunt-beaked birds, and these from the birds that have a pointed beak here. And then we color. So these are, I don't remember, but it's hundreds of SNPs over this region here, maybe 1,000 even. And we color them. So B for blunt is red, P is blue, and the heterozygous are yellow. So you see that all blunt birds with blunt beaks were homozygous BB for all SNPs across this region, except one, which is heterozygous across the whole region. So apparently 2 types of haplotypes. If you go to the pointed beak, you find they are homozygous P instead, except 2 birds which are heterozygous. But what was very interesting for us was these guys, Geospitza fortis. That is the medium ground finch. And you see one is homozygous blue, one is homozygous red. So one homozygous blunt, one homozygous pointed. What that tells us is that there's segregation for this haplotype within this species. And that was interesting because the fortis is the Darwin finch which showed the biggest variation in beak shape. And this also, because you could say it's a bit fishy to make this comparison across species, but these allow us to make a contrast within species. So we did this and this is birds from the same island, and now we just took some of the SNPs and genotyped them and got this beautiful linear relationship. So pointedness, so the ones which homozygous for pointed have a more pointed beak, and you see this. So this is a Mayo QTL affecting beak shape in the Darwin's finches. So the ones which are BB here has the same genotype as this guy have, whereas this has the one which these species have. Then we did a phylogenetic tree of this analysis, and what I thought was fascinating was that if you do a phylogenetic tree of this region, they don't cluster according to species. They cluster. So the blunt is all here in this. So there's different species. So this Magnirostris, Conirostris fortis, whereas fortis is also found somewhere down here. Here you have fortis instead. So when they have the pointed haplotype. Another thing which I think is interesting is when we try to estimate how old these haplotypes are, like we did with the Ruff haplotypes, then we come to that the split between these haplotypes seems to be something like 900,000 years. So why should 240 kb be maintained over such a long period of time? So if it was just one mutation that affected the beak shape. And what I think is the explanation for this, so one could be that it is an inversion that is keeping this. We have no indication that there is an inversion there. But what I would like to say is that what we have been doing, what we did when we was on Galapagos was that we brought nanopore sequencer with us. So we have done this nanopore long read sequencing. So now we will go through and the preliminary data say that there is no inversion in this region. So it's kept as a large haplotype for a long evolutionary period of time. And I think the explanation is what I told you before, is the evolution of haplotypes. So I think there's been a mutation hitting this region, and then you have accumulated additional mutations that make it to a blunt beak haplotype versus a pointed beak haplotype. And that this haplotype have played a role for the evolution of beak shape throughout the evolution of Darwin's finches. So if we look at this, so what we took this 240 kb region, and then we looked for the most mutations or SNPs hitting the most conserved sites. So we found 4 is sitting on, has been classified as transcription factor binding sites. So remember now, this is a transcription, homobox transcription factor, which often have a very distinct regulation. And we also find 4 mutations in the coding sequence. 2 are synonymous, but 2 are non-synonymous. I also would like to pay attention to the color here. So red means that the mutant is a derived allele on the blunt beak. And what you can see is that the derived allele is present 7 out of 8 is a derived allele on the blunt haplotype, which make a lot of sense because we know that the ancestral species had a pointed beak if you compare with the most close relative on mainland South America. So the evolution of the blunt beak happened on the Galapagos because there were many open niches that for food resources they could utilize. So we think that the blunt beak evolved on the Galapagos, and that fit that we see an enrichment of derived mutations on the blunt beak haplotype. So this shows how extremely well conserved this transcription factor is. So here you're looking, so the first line is Magnirost is blunt, and then Conyrost is pointed. Then you have other birds, then you go into lizards, turtle, human, other mammals, dolphins, and you have fish at the bottom here. And you could see how extremely well conserved, so this is the homobox domain, how extremely well conserved this protein is. And look at this now. So this is one of the substitutions. So it's isoleucine and it's valine on the, so the blunt beak has valine and the pointed beak has isoleucine. It's a conservative shift, but look how conserved isoleucine is there. There's only 2 fish, but even zebrafish has the same isoleucine as human have, as the pointed beak Darwin's finches have. But the blunt beak Darwin finch differs. And there's also a mutation here. So these are the 2 missense mutations. And if you now compare Charles Darwin with the sharp-billed ground finch, you find that in the most conserved part of the ALX1, there's only two amino acid substitutions over 300 million years. But if you now compare the sharp-billed ground finch with the large ground finch, there's also 2, this is like 1 million year, and this is 300 million years. So if you take the whole sequence data, add other birds, you see there's an accelerated evolution at this locus in the Darwin's finches, suggesting that I'm willing to bet that at least one of these missense mutations is causal and affect function of, and I could even think that when such a missense mutation happened, it could have some deleterious negative pleiotropic effect, which changed an important homobox transcription factor. And secondary mutation could compensate for a defect in the first. But if the first mutation make the beak more blunt, so they could feed on seeds that other birds cannot do, that could have a big selective advantage in an environment where there is a shortage of passerine birds that compete for seeds, for instance. So with regard to beak morphology, I think we have identified the 2 major loci that are contributed. So we have one ALX1, which is beak shape, and in this case, I'm convinced that ALX1 is the gene that is affected by this selection signal. HMU2 is also this big block. Here the haplotype is approximately the same age, but it's 500 kb, containing multiple genes. The reason why we call it the HMA2 locus is that HMA2 has a well established role in controlling growth, and this is beak size. But I'm not sure that HMA2 is our gene, but it's a haplotype including HMA2. And what we have there is this control beak size, and this contain beak shape. So then you can go from a warbler finch, which is homozygous for the small allele at HMA2, and the pointed allele at LX1. This one has the large allele, so a larger beak, but pointed, that is the common cactus finch, and the difficilis. And this one, the large ground finch, has the large allele at HMA2 and the blunt allele at LX1. So if you go through the species, you could sort them according to that. A very interesting story with this locus is that there was a severe... So what you need to know about the Galapagos is that the climate changed very much from year to year. They very much... So Galapagos is on the equator, but they're very much affected by the El Niño effect. So you could have years of drought followed by years where they have a lot of rain. So 2004 to 2005, there was a very severe drought, which means there was new seeds, there was no flowering coming up. So the birds have to survive on the seeds that were available. And that leads to very strong competition between these two guys. So this is the large ground finch, this is the medium ground finch. On the island Dafna, the island where the Peter and Rosemary Grant spent 40 years. Not the whole time, but a couple of months every year for 40 years. And so they were competing for the available seeds, and what they were able to document, so this is a science paper, is that they show that, as I mentioned to you, that there's a variability in beak shape and beak size among the medium ground finch. So in fact, what they observed here was that from one generation to the next, there was a reduction in the beak size. So these guys got smaller beaks, and the reason was that birds, the medium ground finch with larger beaks, did not compete well with these guys. But those with smaller beaks could feed on the seeds which were too small for these guys. So they escaped competition. And that in ecology is considered a very important part of speciation because it leads to character displacement. So the species sort out in what they are feeding on. And then what we know is that these are fixed for the large allele at MG2, whereas these are segregating at this locus. And the beauty is that Peter and Rosemary have ringed all the birds, they collected blood samples, and we could see which survived and which did not survive this drought. And it turns out that there is a very striking change here. So if those were medium ground fish that were homozygous for the large allele, only 30% survived. The heterozygotes, 53% survived. And homozygous for small survived, 74% survived. And so this locus explains as much as 30% of the selection response during this selection. So one locus explains 30% of the change in beak size from one generation to the next, which is fascinating. Now to something completely different. So just to give you a feeling for the organism, is a picture of Norwegian herring in this. So Atlantic herring, one of the most abundant vertebrates on Earth. So we estimated that the breeding stock of herring in the Atlantic and the Baltic Sea together is on the order of 1,000 billion individuals, 10 to the 12. And this was the place where I started to do research, in fact, and didn't find any genetic difference between populations, so I gave up at that time. But I would say that I've argued that herring can be a model organism for genome biology, and the reason is that it's a near ideal population structure for being a vertebrate. Huge population size, random, so that when they are mating, they have this broadcast spawning. They're aggregating. Millions or billions of fish are aggregating, mixing sperm and egg, and really panmictic matings. Very high fecundity. A female can do 20,000 to 50,000 eggs in one spawning. Natural selection, of course, very effective. And this is what I think is really important, that genetic drift at selectively neutral loci is minute. So if you take neutral markers and genotype them, you cannot distinguish herring from the Baltic Sea, which is brackish, from herring catch in Newfoundland. And then they have interesting ecological adaptations. One is different marine environments, including the brackish Baltic Sea. So very few marine fish is able to go into the brackish Baltic Sea, but herring can do it. There's also difference in spawning time. So some breed in spring, other in summer, others. So spring and autumn is the major, but it goes into the summer as well. So something that we have observed, so it's an Leif paper from 2017, is that the Atlantic herring has a very low mutation rate. The reason why we wanted to do that was that although it has this enormous population size, the nucleotide diversity is not as huge as you could think. So human have, if you sequence yourself, you will find one SNP every 1,000 base pair. If you sequence a herring, you find a SNP, 3 SNPs every 1,000 base pair, but which is much higher than human, but not huge if you think about the enormous census population size. So we wanted to look at, and it turns out that herring has a low mutation rate. So it's the lowest among any vertebrates so far, and it's to the order of, so we did it by sequencing parents and offspring from pedigrees and counted the number of, and it's almost an order of magnitude lower than in humans. But what I think is very important then, because of this enormous population size, so in each generation, each nucleotide mutates thousands of times. So they have this enormous opportunity to test different sequence variants at each position throughout the genome. So we published a genome assembly in 2016 in eLife. So it's about 800 megabyte base pair in size. 20,000 genes, GC content normal. So it's a good assembly, but highly fragmented. So this was Illumina mate pairs, different size of mate pairs, and so on. But we just on the way to release the version 2, we have done PacBio long read sequencing. We have a high-resolution linkage map. We have a high-C, and which makes it into a chromosome-based assembly. And so now, the number of chromosomes in herring is 26 chromosomes, total size about 800 megabases here, and quite equal size. So most of the chromosomes are in the range 29 to 37 here. So one is 24, 17, and 114 here. And we also have this estimate of recombination rate across the. So this is in fact based on linkage disequilibrium patterns. So we see this very, very striking that we have regions with high recombination and regions with very low recombination. So very uneven distribution where recombination takes place in this species. And it's present both in the linkage disequilibrium pattern and in the meiotic data that we have. So what we have done with the herring is that we have done a comprehensive screen of genetic diversity. So we sequenced, so we have 67 populations across the distribution. So we have from Canada, we have from Iceland, we have from Greenland, we have the Baltic Sea, which is brackish. You have 3 per mil salinity here, going down to like 12 down here, and here's 35 per mil salinity outside here. We have also done individual sequencing as a complement to the pool. So pooled sequence is a very cheap method to rapidly generate allele frequencies. So if you do the pooled sequencing data, so this is a quite interesting phylogenetic tree. So this is all the diversity or the difference between all populations in the Atlantic, including Baltic Sea and the Atlantic Ocean, whereas Pacific herring show more differentiation between populations from different regions here. So this is a fjord in the north of Norway where you have hybrids between Pacific herring and Atlantic herring. So what happened was that during glaciation, Pacific herring and Atlantic herring is completely separated because there's ice in the where the Arctic Sea is. But when it's warmer apparently what have happened after the last glaciation is that Pacific herring colonized the Arctic Sea all the way until the Atlantic Bay. So they meet in North Norway, and where they apparently hybridize. So we're currently analyzing this. But I mentioned to you that my first project as a biologist when I was an undergraduate was to study genetic difference between populations of herring. When you see this, you realize that it was, if you just take random marker, your chance to find difference between populations is very, very small. But if you zoom in, you start to see, so this is in fact Canadian herring, and this is herring from Norway, Iceland, and so these are herring from the Baltic Sea, and these are populations around the British Islands, and so on. So it's, and I would argue that most of the differences you see here between populations is due to adaptation. Neutral markers tend not to show difference between populations. So then is this question, how polygenic is ecological adaptation in a species with a huge breeding population? So, you know, I did this work when I was a student. I took 14 genes, no difference whatsoever. Every population had the same alleles, same allele frequencies. You went from Baltic Sea out in the Atlantic. And you know, so I was thinking about that for 30 years. I'm waiting for them to invent next generation sequencing, because now is the time to answer this question. And my idea was that this would be an advantage if there is no differentiation at neutral loci, because then you would see selection taking place. But my worry was that it's so polygenic that we don't see any signal, because if the mutation happens on many different haplotypes, it will be just, there's so many loci that contribute to adaptation to a different marine environment that is just a smear across the whole genome. But that was not what it was. We saw very distinct signals, and maybe this looks like a smear, but what I could tell you is that what you see here, so each dot is one, so this is when we compare Atlantic versus Baltic. This is when we compare spring versus autumn, and you could see the p-values here, 10 to the minus 100. So very, very strong signals, and it's only like 2% of the SNPs that show this strong differentiation. So the great majority of SNPs is just around 0 around here. So you see that very beautiful if you zoom in on the chromosome. But this is, so this looks really messy, and that's because we have a fragmented assembly. When you have a chromosome-based assembly, everything looks much more beautiful. We turn them upside down to make it more complicated to look at as well. But then you see this fascinating, clear signals of selection across the genome. So we have hundreds of signals that is related to what happens when herring adapted to the brackish Baltic Sea. If you compare spring versus autumn, you once again see these beautiful, striking signals. So much fewer signals here than there, which this is adaptation to the Baltic. This is just a difference between the spring and the autumn spawning herring. But look at this signal here. Here you have tyrosine-stimulating hormone receptor, which has a key role in photoperiodic regulation of reproduction in birds and mammals, apparently in fish as well. So what we did recently was that we looked at populations around the British Islands. This has not been published yet, so we are just on the way to put that together. And once again, you see this, if you compare the populations around England and Ireland, Scotland, versus other populations in the Atlantic, not the Baltic, we skip those now. And you once again see some very, very striking signals. And this is very suspicious to me when you see this one. This is a very thick signal. So, and if you look, now we look at individual SNPs here from that region, in fact. So here you have, so each column here is one SNP here, and then we color them. So the one, if they're 0 means that there's 0 frequency, and 1, so what you see here is that these populations up here has a completely different haplotype compared to these ones, for instance. This is Norway, and this is Canada, and in general, so this is a very, very, and the interesting thing here is that it doesn't matter which SNP you pick here. You see the same pattern, right? This is 8 megabase region, completely different from what we've seen before. So this is very, very strong, suggesting that it's an inversion, segregating in the Atlantic herring population. And so this is a contrast where we do spring versus autumn. So this is in fact the locus which probably have something to do with the difference between spring spawning and autumn spawning. This region has nothing to do with spring or autumn spawning. It just happened that when we do this contrast, the frequency of these two haplotypes differs between the two groups. But what I wanted to show you was that you see this block again. You see the same signal across this 8 megabases region. And we didn't see it so clear before because the assembly was fragmented. It was 4 scaffolds. But now when you see the 8 megabase going as a block, it becomes very clear. So then we have done SNP typing on about 1,800 fish. And then now you could sort them. So these are the heterozygotes. And then you see, if you look at linkage disequilibrium here, you see beautiful linkage disequilibrium across the region, as I showed you before. If you do this now on the sort of southern homozygotes and northern homozygotes, the LD goes away. Because you could say there's something else which suppresses recombination. So you see LD also in these homozygotes. But that is not the case. It looks like you have normal recombination going on when they are homozygous for one. So I think this is a very strong indication that here we have a true inversion that is involving ecological adaptation. Then now we could then measure the frequency of this, and this is done from individuals. And we call this northern and southern because one type is very common in the south. So blue means they are the southern dominate. Sorry, it's a little bit confusing here. Yeah. So the frequency of northern allele, 100% is occurring in the north. So this is in Newfoundland. You have Norway here. You have inner Baltic here. But as you go out here, you see, and you should remember now, if you go through 6 million SNPs, that 5.8 of those will show no difference. So this is highly significant. This is definitely a locus under selection. Our guess at the moment is it has to do with water temperature, because this is where the Gulf Stream coming from the Gulf of Mexico are hitting Europe, and this is the warmest part where the herring is breeding. So we think it's most likely related to adaptation to water temperature at spawning. So I think for me this is a very important figure from one of our herring papers. So what we're doing here is to look. So since we have hundreds of loci in the selection, we could ask the question: what type of genetic changes are enriched among those showing differentiation between populations? So what we do is that we classify SNPs as, I call it delta allele frequency. So this is the delta allele frequency between Atlantic herring and Baltic herring as a group. So now you see, so this the black curve is the number of SNPs. So you see the majority of SNPs here show very, very little difference in allele frequency between Atlantic and Baltic. So very few SNPs comes out here. You start with 6 million and the fraction is coming here. Then we ask the question, if you classify SNPs according to a function, what is their relative frequency in these different bins? If they occur at the same frequency in one bin as in the rest of the genome, you will get an M value of 0. So if there's no difference in their occurrence in the different bins, you get a flat line here. If they deviate from that, you will see, so interesting, intergenic is under, so these are the ones that show difference between Atlantic and Baltic. Intergenic are underrepresented. But look at this beautiful red, which is coding. Non-synonymous changes is highly overrepresented among those that show differences in allele frequency between Atlantic and Baltic. And if you compare that, so even synonymous saw this pattern. But in this case, I would say, this, you should remember, this is a logarithmic scale. So the signal is so much stronger for the non-synonymous than the synonymous. So the change in synonymous could be hitchhiking on the non-synonymous. So I think this is a very, very strong evidence that changes in coding sequence has contributed to ecological adaptation in the herring. Does that mean that coding changes are more important than regulatory changes? No. So you see for 5 kb upstream, 5 kb downstream, and also UTR, you see signals here that they are also enriched in this area here. So the reason why that is not as strongly enriched is that there's a very poor annotation. I just say they are 5 kb from upstream or downstream of coding sequence, whereas in the non-synonyms, I could tell if it's changed amino acid or not. So the day we can have better annotation, I think that, but this is a beautiful data where we could test which type of genetic changes has been most important during herring adaptation. So I will give you one example where I think we have a causal mutation, which I think is amazing, going from sequencing fish to nailing it down to a causal variant, and that is rhodopsin. So here we compare Atlantic versus Baltic. So the strongest signal we have is this gene, in fact. So this LRRC8C is an ion channel. We have 4 missense mutation. I'm sure that is also causal variants. But we're waiting for the experimental data to validate. This is an ion channel expressed in sperm, in fact. But rhodopsin. So as you know, rhodopsin is found in the rods. And the critical thing with rhodopsin is that it's critical for vision in dim light. You have retinol, which is where light absorbance takes place, which is held by this G protein coupled receptor. So if you zoom in on this region, then this is the signal you're comparing. So this is what it looks like with each dot here is one SNP here. So you see it's completely black underneath here. And then comes this beautiful signal. And the red line here is where the rhodopsin locus is. So the next thing we did, so we decided to look only of the SNPs which are found only in the Baltic. And then you get an even more sharp. Now we zoom in around rhodopsin here. And you could see that we have in total 100,000 SNPs in this area, which is unique, segregating in the Baltic, present in the Baltic. 1,000 of those are missense mutations. But the majority is down here, low frequency. But you have 2 missense mutations, one up here. And in fact, so this is very interesting. It looks like you have this derived mutation, and then by recombination, they show less and less signal than when you move out from rhodopsin. But this is an interesting thing. This is within this pattern here. This is a second missense mutation. So this is phenylalanine to tyrosine at 261, and it's isoleucine to threonine at 213. And this is what it looks like if you look at the allele frequency of this missense mutation. So if you start in the Atlantic and then you move towards the Baltic, and the further you go down here, you have some population which are completely fixed for tyrosine at this position. This one is not fixed yet, so it has like a 25% frequency, maybe 25-50% frequency. Haven't gone to fixation in any of the samples we have. So then is the question, is this just hitchhiking on, so selection act on this one and this happened to hitchhike on it? Then you should remember about 90 billion Baltic herring in the Baltic. So there's a lot of chromosomes there. So for something to hitchhike, this mutation must hit very early in the expansion of this haplotype. So I think that suggests that maybe it's in fact also under selection. So then you could do amino acid alignment, and you take fish here, and if you go to human, you have phenylalanine. All birds and mammals, I think, have phenylalanine here, but a couple of birds have tyrosine here. So then is the question, is it just that this is a neutral SNP that is allowed, it's tolerated at this position? You could compare phenylalanine with tyrosine. So the only difference between these two is a hydroxyl molecule there. So if you're not a chemist, you think it's a tiny difference. It's probably neutral. But the thing is that rhodopsin is a molecule that's been studied very well. There's a crystal structure from bovine rhodopsin. So I added some cattle genetics for Harris. So bovine rhodopsin, the structure here. And here you have phenyl 267. And you could see it's very close to retinol. And this data suggesting that it may. And look now, if you so this is in fact modeling the herring sequence on the bovine rhodopsin structure. So look here now, this is phenylalanine, this is tyrosine, this is this hydroxyl molecule, and it's pointing towards retinol there. So it's in a perfect position to interact with retinol, where photoreception takes place. What is interesting, which I didn't know before, was that there's a clear difference in the photo environment if you go from the Atlantic or from an ocean into a brackish water into a fresh water. So this is the aquatic elimination spectra. So you go from open Atlantic, deep water, less deep water, into the Baltic, which is not as deep, and what you see, and then you go to a freshwater lake, and you have this red shift. So there's more abundant red light in a freshwater lake than in the open ocean. And the Baltic is somewhere between the ocean and the lake, and it's probably things that is washed into the water that absorb light and how light penetrate into the water. So I would say there's conclusive evidence for the functional significance of phenylalanine to tyrosine at this position. So, and this was a study published already 92 in GBC. The reason is that we have red opsin and green opsin in our eyes for color vision, and they differ at this position. And the question was, is this one of the residues that explain why red opsin and green opsin have different absorption spectra? So they mutated this site, and what they show is that if you go from phenylalanine to tyrosine, there's a red shift in absorptions of 10 nanometer. The Mexican cavefish carry the same mutation as the Baltic herring, in fact. So the Mexican cavefish are forms that have some vision and some which are completely blind, but they have tyrosine as disposition. So they made 95, they did an experiment where they expressed this protein, and then the other way, and they get the blue shift. If you go from tyrosine to phenylalanine, a blue shift of 8 nanometer. And then there's another group which did publish 2007, and then they looked at absorbance of the retina from herring catched in the Baltic versus in the Atlantic. And what they show is that the absorption spectra for the Baltic herring is 10 nanometers shifted compared with the Atlantic herring, which fit perfectly with what happens if you make this change here. So I'm absolutely convinced that this is a causal variant. So what is the idea? So that means that the Baltic herrings see better in the light conditions in the Baltic than what the Atlantic herring do. So if you look now, what I didn't know either was there's more than 1,000 species. The sequence from rhodopsin from more than 1,000 fish species already, because there's been special interest to study the structure function of rhodopsin. So if you start looking at residue 261, out of 1,000 fish, there's only 2 amino acids at this position, phenylalanine and tyrosine. That's the only, and that's, if you take the sum here, 1,000 fish has phenylalanine and 143 have tyrosine. So this is the Baltic, this is the Atlantic herring. There's more variation at position 213, but I would argue that it looks like what happens that when this mutation happens, the chance that you also mutate to threonine, so this is what the Baltic herring has. They have, this is the majority, but some of them have this mutation also. And if you now see the proportion among species, you see that only 10% of so this is the ancestral, only 10% have threonine if there is phenylalanine at this position, whereas 1/3 of the fish species has threonine position at this position when they have tyrosine at this position. So what that means is that the likelihood to fix this change differs depending on which amino acid you have of this. So once again, going back to this message that the chance of evolution differs depending on which allele you have to start with. So what I think we have here is an amazing example of convergent evolution, because what it turns out is that there's a number of species that have this change have happened. And here you have the Baltic herring, but you have the Mexican cavefish, the northern pike, there's 2 species of sculpins, the spotted gar. All freshwater fish have this tyrosine, and Baltic herring, so far it's the only marine fish, but we are going through this So we're waiting to get the complete list of the habitat of all the 1,000 fish species to see. But the one we have done by hand, there's a perfect match that the freshwater fish tend to do the tyrosine, whereas the marine fish has phenylalanine like birds and mammals have. So I think it's amazing to see convergent evolution happening at a single amino acid residue. And I will end by thanking. So the Ruff story and the Darwin Finch story I told about has been published, but the herring work is on the way to be published. The assembly, Mats Pettersson has been involved, and we have collaborated with people from different places, BGI, for instance, for the assembly, and herring population biology. And Nils Riemann was my former supervisor who kept these samples in the freezer for 30 years before I was ready to take care of them. Thank you. Sorry for being late. I exhausted you. Yeah.
In the region that you found in the Darwin's Finches, you say you didn't think it was an excursion. What do you think is suppressing?
I mean, you could say if there are multiple changes in that region which affect fitness, so recombinant haplotypes may have lower fitness and is then lost for that reason. If that happens, you would expect that also you have selection for reduced recombination. But it looks like, in fact, that this ALX1 is in a region. So in fact, ALX1 and AHO2 are linked by quite far away from each other on chromosome 1. But it looks like they're both in a region of low recombination. So maybe that has added to building up haplotypes. So. You know, for us, it's difficult to say which large effect they have, you know. So what we see is what have happened with, so we see that the herring has adapted to living in the Baltic. And what we could then see is which loci have showed changes in allele frequency. But we really don't have a clue how strong effect they have. Because, you know, we can't really measure fitness from different phenotypes in this case. So,
the adaptation is associated with known phenotypes that we can measure.
No, so what I think we have to figure out what the difference. So rhodopsin, you know, vision. I mean, I think it's very clear that here we have a link between a function and the change, a difference in the environment. So I mentioned this other locus. So that is this LRRC8C. This is called this is a VRAC ion channel. So this volume-regulated ion channel. So what happens? So these are found in all vertebrates basically. So what it happens is that when the cells swell, so that the volume change, these are activated and start to pump natrium, sodium out of the cell, and this particular there are 4 isoforms in vertebrates, and this specific isoform is the only one expressed in the sperm of the herring. So we have a collaboration with the group, and you could think of, so what happens with the sperm, you release it into the seawater, and it should be activated by seawater. And of course, there's a big difference to spawn in brackish water versus in marine water. So now we are testing this ion channel. So I think we have to go, so locus by locus, basically, to find out what is going on here. But it's difficult for us to measure fitness in the Baltic Sea. So I would say, but the evidence for selection is outstanding. And the reason is that, so what I think is going on is that you have little genetic drift because you have huge population size, but then you also have So they have homing behavior. They tend to go back to the same spawning area, but then I'm sure there's gene flow between places. So.
You dated the inversion 4.5 million years approximately, right?
About 4 million years.
About 4 million years. So do you have an idea if that predates the speciation event, or do you think it happened after?
So that was one of the things we thought about. Is could we see but there's no species. So the closest relative is like 5 or something million years, and they don't have this leg. So I think the leg was the starting point. So the reason why this alternative male were attractive strategy was because the dominant male have to spend energy on fighting, defending territory, open up. That is advantage to have an alternative strategy. So.
You don't believe it's an example of macroevolutionary change associated with the rearrangement?
What do you mean by?
So, I mean, this is a dramatic phenotype.
Yeah.
And if the inversion is responsible for the phenotype, then that's a very big change.
I'm 100% sure that the inversion is not doing everything.
Okay.
Absolutely, and we have this. I mean, it's very striking. So this inverted chromosome has shrunk. It's a bit smaller, and there's a concentration of deletions around that gene, which has a role in testosterone metabolism. So what I think is happening is that the first inversion happens, gives a phenotype, and then mutations that increase the fitness of that will accumulate on that chromosome. So what I would say that there is an interesting, I think we have an interesting story from domestic animals. So rose-comb chicken, they have an inversion, but that is a recent inversion, I would say, which happened after domestication. 7.4 megabase inversion, what it does is it moves a transcription factor into a new chromatin environment. So you upregulate the transcription factor in those cells where the comb is developing. It's also breaking another gene. But that is like the only phenotype you see in those. So change the expression of one gene and it disrupts the effect of another gene. And so I think often when you have inversions, what's happening is that they change the chromatin interactions and around the breakpoints. It could even be outside because you move regulatory domain to a region outside the inversion. So I think that gave the first phenotypic effect, but then you accumulated changes on top of that. So.